Good evening, everyone. My name is Joel Jonathan from Bachelor of Bioinformatics. My supervisor name is Dr. Ropaning Sulo. The title of my thesis is Genetic Diversity and Phylogenetic Study of Aspartic Protease in Nepenthes Species from Asian countries. The problem statement. Nepenthes is widely distributed around Asia continent. It can be found in Malaysia, Borneo, Cambodia, China, Philippines, and Indonesia. Due to the different ecology needs, this plant undergoes severe evolution and mutation to survive the environment. Different species of Nepenthes have different morphology due to its scarcity distribution. This thesis will help researchers to understand the difference of morphology and gene sequence of aspartic protease of 15 different types of Nepenthes. Due to lack of research on aspartic protease structure in Nepenthes, this study will predict 3D structures of Nepenthes raja, which will be used for the future reference by researchers. The objectives to identify the similarities and difference among 15 different aspartic protease sequences from 15 different species of Nepenthes around Asia continent, to analyze the phylogenetic tree of Nepenthes from neighboring countries, to predict the 3D structure of aspartic protease from Nepenthes raja for further research. The methodology. The first step is NCBI database search. The search of 15 sequences of aspartic protease from 15 different species of Nepenthes in Asia. The second is multiple sequence alignment. Multiple sequence alignment will be done using MEGA with the 15 search aspartic sequence to find the relation of the species of Nepenthes. This multiple sequence alignment is done to find the relatedness of the species. Protein 3D structure predicting is a third step, using the sequence of aspartic protease in Nepenthes raja to predict 3D structures using eye taser. Next is the result. Table 1 shows the data of 15 sequences of aspartic protease from 15 types of Nepenthes in Asia, which is obtained from the NCBI database search. This species is from Malaysia, Cambodia, China, Indonesia, Borneo, and Philippines. Next result is Figure 1, Phylogenetics 3. Figure 1 shows the Nepenthes raja from Malaysia and Nepenthes dentaculata from Borneo are very closely related and may have the same ancestry. We can, however, speculate that Nepenthes ventricosa was the ultimate parent of both Nepenthes dentaculata and Nepenthes raja due to its increased diversity. Nepenthes mirabilis from China, Nepenthes alata from Philippines, and Nepenthes gracilis from Cambodia are far more similar to other Nepenthes species due to its greater geographical distance between them. Nepenthes sumatrana from Indonesia and Nepenthes stenophila from Borneo are very closely related and may have a common ancestor with Nepenthes tobaica from Indonesia. We can, however, speculate that Nepenthes tobaica from Indonesia was the ultimate source of both Nepenthes stenophila and Nepenthes sumatrana due to Nepenthes tobaica's increased diversity. Nepenthes thai from Thailand and Nepenthes alba from Malaysia are closely related, as are Nepenthes longifolia and Nepenthes sagani from Indonesia and Malaysia, which may have common ancestor. The next result is the 3D structure. Structure 1 shows the most confident predicted 3D structure of aspartic protease in Nepenthes raja with C-score of -1.26. This structure is the most confident because the C-score is closer to 1 than the other structure and the number of decoy used in this is high, which is 600, with the cluster density of 0.01005. This protein structure is predicted based on the sequence of proteins in Nepenthes raja. Structure 2 shows the second most confident 3D structure of aspartic protease in Nepenthes raja with a C-score of -2.32. This structure is almost similar to structure 1, but the number of decoy used is 468, which is lesser than the previous structure, and it is stated the cluster density is 0.0349. Next is the structure 3, shows the third structure predicted for aspartic protease enzyme in Nepenthes raja with a C-score of -3.19. This structure is lesser similar to the 3D structure 1 and 2 because the shape of the structure. The number of the decoy used for this structure is 222, which the cluster density is 0.0146. Next is the structure 4, shows the fourth predicted structure of aspartic protease in Nepenthes raja with the least confidence score, which is -3.17. This structure is confidence level is higher than the structure 3 with a C-score negative 3.19. But due to the number of decoy used is 212, which is lower than the structure 3, and the cluster density is 0.0148, this structure is predicted as less confident. The final structure is structure 5, shows the least confident structure of aspartic protease in Nepenthes raja because the C-score is negative 2.94 with the cluster density 0.0187 with the lesser number of decoy which is just 188. Conclusion. The tropics are habitat to Nepenthes, which is found across the tropics but is most abundant in Borneo, Sumatra, Peninsular Malaysia, Philippines, and New Guinea. It reaches New Caledonia and the island of Pines in the east, the Seychelles and Madagascar in the west, the York Peninsula in the south, and an isolated area on Cassia Hill in India and Indochina in the north, but not in Burma. This research purpose is to identify the similarities and difference of aspartic protease in 15 different species of Nepenthes in different Asian countries. Multiple sequence elements done, and the conserved site and a variable site is identified in the process. This makes us to understand difference in the distribution by morphological and molecular data using aspartic protease, which can be found in the digestive juice of Nepenthes. With the data obtained, the researcher predicted the protein structure of the aspartic protease as a reference for future researchers. From the phylogenetic analysis, we can figure out the ancestor species for the Nepenthes. From the phylogenetic tree, all the objectives of this thesis are obtained successfully. Recommendation. This research makes some key recommendations to boost the agriculture production, particularly the Nepenthes raja plant species. These suggestions are based on the suitability of the raw material equipment, expenses, and federal law. This thesis will focus on identifying the similarities and difference between 15 different species of Nepenthes from various countries, as well as predicting the 3D structure of aspartic proteins present in Nepenthes raja for better comprehended the protein sequence. This is a group of personnel who are strongly advised to read and discuss the review. First is researchers who are doing research about Nepenthes raja. Second is company that wants to implement and yield Nepenthes production via plant tissue culture, industry, and agriculture. Third is farmer that searching the method to grow their breeding technique. Thank you.